Our next speaker is Dr. Ramesh Sagili from Oregon State University, and he'll be giving us Varroa research updates from Oregon State. And welcome, Ramesh. Thank you.
Thanks, Steve. Yeah, hope you all are doing well, staying safe, and eagerly waiting for this disastrous 2020 to get over. Can you hear me, Steve?
Yes.
Yeah, sorry.
Okay, cool. Yeah,
we can hear you.
I didn't get a response from Steve, so I thought maybe he's gone. Okay, so what I'll do in the next 40 minutes is go over some of the Varroa research that we have been doing for the last couple of years, I would say, and then see if I have some time for questions at the end. Okay, hope this moves. So before I start, I just wanted to acknowledge all these people. I know I don't have all those people in the lab right now, but I'm showing some of our previous worker bees as well. Without these wonderful people, what we do here at OSU is not possible. So I really want to thank them before I start. And here I have the latest ones that are currently in the lab. Probably some of the Washington State beekeepers. We do interact them quite a bit, so probably they have seen most of them. So this is the overview. I'm gonna spend some time on some basic information on the population dynamics. I know Randy was getting some exciting questions from the audience about how long the mites can stay on the bees and or survive on the bees. So I'm not gonna go in too many details, but we did a population dynamic study, which I'm gonna go briefly today. And then I know there is a huge interest in oxalic acid vaporization, at least for the last 3 or 4 years. Since it was registered. So so we did a quick study on looking at brood mortality with oxalic, and then I'll go over one study we did last year looking at amitraz resistance, or resistance probably is not the right word for this study. Maybe we can say efficacy study of amitraz. So here I have, for those, bear with me, those veterans here, but for the new beekeepers, I know there is a significant audience that might have started bees last year or this year. So I just don't want to lose them. So I may go into some very basic information here. So bear with me. So here is what I know. Randy gave an excellent talk and you heard about all that mite stuff before. So this is phoretic phase where you're seeing mites on the bees, and then the reproductive phase is the one which probably we don't think, especially as new beekeepers, probably we don't pay attention. We are just looking for those mites on the bees, right? Looking at how many mites we have in our alcohol wash or sugar shake and all those methods that we look for sampling. So I want to focus more on the reproductive phase. Like how many mites do you think are there hiding in these cells, right? If you read books or past literature, yeah, it says sometimes even 80% of the mites are hiding here inside the cells, or 20% are in the phoretic phase. So let's see what we found in one of our study. And it's very variable. Don't take it very seriously when I give you a number. This is just based on our one study in September. So I'll go over that in a minute. So for those, especially for those new beekeepers, I just wanted to refresh your memory of the Varroa mite life cycle. So here is a hive. So you all know the bees are in the the mites are in the phoretic phase. They are getting into those appropriate cells based on those larval cues, the pheromone cues they get on the right age larva. Here you see the mite is hiding underneath until it is capped. And then she comes out and starts laying eggs. And usually it's shown that the first egg is a male. Then she may lay 5, 6, 7, depending on whether it's an drone cell or a worker cell. So here you see the progeny, and we use different terms because mites are not insects. So we use those terms nymphs. And then once the time arrives for the bee to chew through the cappings and gets out, and here you see those, at least one female mite offspring will come along with the foundress mite. So that's basically what our Varroa mite life cycle is. I apologize for making it too simple, but I think just for the first timers, it's easy to understand. So I'm not going to talk about mite. I know your Washington State Beekeepers group is an amazing one. You probably are related to your regional groups as well, and you already know what kind of damage mite does. And Randy gave a great talk on some of the viruses and how the damage is done. And even Jennifer had some slides on the mites and the mite problems that we have. So I'll not go over all that stuff, but just to just to give you an overview, the parasitic mite syndrome, probably you hear about that a lot. So see some deforming virus bees. There'll be some larvae that are actually not because of bacterial diseases, but because of the Varroa mites. And you see a lot of this spotty brood with some of these chewed mites, probably hygienic behavior, we don't know. So here is a question for you. So I'm asking you, what percentage of mites are in the capped brood cells and what percentage are on the adult bees, which is the phoretic phase? I just want you to think. I just threw out some numbers here. Probably the answer isn't here either in one of these, but I just wanted you to just think about because we tend to not think too much about what's going on inside the cells because we don't know exactly what's going on. Okay, so this was a study we did, I think 2018. So we were trying to estimate the phoretic and reproductive mite populations in these colonies. And we also compared from the same colonies because we were collecting data. So we did some meticulous data collection for alcohol wash, sugar shake, and sticky boards. But then I saw Randy talking about all these. So he gave an excellent talk on these methods. So I may not probably go into those. I actually deleted those slides so that I'll save some time. So I'll focus on this part, estimating phoretic and the reproductive mite populations in the colonies. So what you're seeing here, there is a truck or lab truck. So we chose very isolated locations. So Oregon State University is in Corvallis. So we have about 40 miles away, there is an apiary that we have. So there is no beekeeper at least around for 5 miles. So we thought, because these hives were supposed to breed for mites, not for bees, right? So we wanted them to be as isolated as possible so that we are not creating problems for a beekeeper around. With robbing, drifting, you all know how mites are spread. So here's a brief description of methods. I may be skipping a lot of things, but you can ask me at the end when I'm done if you are not clear on what we did. So we used only 6 colonies. We didn't want to kill too many of the hives for this study. And you will see and appreciate how laborious these studies are if you want to look into each of those cells in all of those 6 colonies. So we had no mite controls from March 22nd on until September 14th. So this is the date when we killed literally all those 6 colonies. So I'll show you how we did that. So the mite levels were monitored throughout the summer. So we had sticky boards. We were looking at alcohol wash methods as well as the sugar shake. So September 14th, as I said, all 6 colonies were killed using alcohol and all the brood frames were collected and they were stored in the freezer so that the students could look at each of those cells at their convenience. So here is just an image on the messy process of killing bees. So you see, I think Randy showed something, a similar structure like this, where you can use a funnel for getting all the bees. So we chose a day when it was relatively cold in the morning. I know summers are not that cold here. So that most of the bees are there. If we are doing in the afternoon, then you will lose a bunch of bees, or a significant population is foraging. So we wanted as many as, so I think we got probably 95% of the bees. So we shake those bees into this funnel, and here is the bucket with alcohol in it. So they will all fall and die. So that's your adult population of the hive, right? So then you can look for mites in that entire bee population of whatever 40,000 bees you have at that point of time. So I don't know if the video is playing very well at your end. I apologize if you can't. But see how that messy process I'm showing the students here are shaking those bees. Each one is looking at those frames to see the brood frames will go to the lab, and all the bees are going into those buckets full of alcohol. Looks like this student is having fun here with the brush. Probably she didn't have enough tasks to do. So that's basically how we collected bees, and these are the bucket full of those bees, dead bees that came back to the lab. And then this is the most tedious process I was talking about, the most painful process. And then you may laugh, like, what's the point of doing all this work? And I think probably Randy will appreciate. I think he has done a lot of this type of things in the past too. But you need someone to do these things to really get a handle on these. When Randy showed you that the model he has for Varroa mites, I think that's how he tries to get those data from different studies as well, so that it would be useful for designing a Varroa pop model or anything that you can think of. So it has its own value, but I'll give you why we did this for, especially for beekeepers that are new. You will appreciate if you understand how many mites are there at a particular point of time in your hives in capped cells. So again, these students are just faking that they're laughing, but I think probably some of them had a bad vision after some time looking into these cells for too long. So yeah, these 6 colonies means you are looking at 20 times 6, right? And then each probably has 3,000 cells to look at as well if they are fully capped. So this is again another image showing you all those pupal stages that were extracted, and you can see those mites on them. And some of them may have 2 mites as well, or 3. Sometimes, if they don't have an opportunity, you can see multiple mites, actually the Fondres mites itself getting into those cells. Again, another image to show you a mite family here extracted from one cell. So again, another image where I'm showing you one of those almost developed bees where the mites are. So here is again, I'm not going in details. I still have to analyze in different ways this data. I just put this for this presentation so that you get some sense on what's going on. So here I'm showing you in September. Again, this may change if you're doing in your own area at a different point of time. Again, it depends on how many bees you have, how the mite population dynamics is going on. So if you're in the valley here in the Willamette Valley and probably in September, this is what you should probably expect in your hives is 41% mites are in the phoretic phase and about 60%, I would say, in the in those brood cells that you're looking at. Okay, so take home message again. So it's about 60% mites in September, as I said, and again, there are so many factors that might be responsible for how many mites you see in the brood cells. But again, just to give you a take-home message from this is, so always think about the hidden mites inside the cells, especially suppose some of you probably use oxalic acid. So if you're using it as a vapor and you're just killing those phoretic mites to some degree with that one application, and don't think your mite problem is gone, right? And it's not, because you have to use multiple times probably. So as new beekeepers, we sometimes tend to think, oh, someone has given me an idea, okay, this is the mite treatment that I need to use. And if you use that product once, it may not work, especially if those are the ones that are impacting only the phoretic phase. So that's what you need to think about always. How many mites do I have in those? Because there are thousands of mites. Maybe there are, if you think 2,000 bees are coming every day, maybe half of them have mites, right, that are coming out. It depends on how many mites you started in your hive. So just I want you to think more about those mites that are inside as well, and use that information for your treatments as well. So that's what I say here. Keep this fact in mind when using my treatments to control Varroa. Okay. So these are some treatments. I know I have not used everything that is available for beekeepers at this point, but these are some common ones, very popular ones like APVAR, which is amitraz. There are formic acids. I think this is now more formic acid. I think someone was asking 1 pad versus 2 pads. So this is again a Formic Pro, which is a popular product for beekeepers to use. And most of them are very effective. HopGuard, we haven't done that much work, but I think HopGuard, at least 5 years ago, we did some work. And when that messy product was there, I think they have improvised their product in the last couple of years, and looks like it's not as messy as it used to be, but we haven't tested the new one, so I don't have much information. Then oxalic is again becoming a little more popular with the sublimation or the vaporization method that beekeepers use. So what I'm going to do is only talk about oxalic today here. So the reason why we wanted to look at this is I've been getting a lot of questions on, yeah, we want to use even when there is brood present, right? Technically, you're supposed to, like the dribble method, people have used it for a long time now, since probably last 15-20 years, very successfully when there is no brood. It's usually November, December, depending on where you live. But vaporization now, some beekeepers say, yeah, I mean, technically you read the label, you're not supposed to use when there is your nectar flow is going on. But some people, if they don't want to extract that honey and they really are more concerned about their mite populations, then they just want to use even when the brood is present. So we wanted to see if it has any impact on the brood. So that's a major primary objective. So here I have those objectives. So we wanted to evaluate brood mortality, both eggs and larvae, when using oxalic vapor. And this is more like a secondary one. Don't glean too much information from the second objective because it was only one oxalic acid treatment. So yeah, it's not fair to compare just one oxalic treatment for a for a for a formic, which is probably, if you're using one pad, it's like 20 days, and if it's only one, then two, then it's 14 days. So I'll go back to that again when I talk about this efficacy part. But this is the most important one. We wanted to look at the brood mortality using oxalic. So this is how you should apply. If you're using oxalic, I really tell hobby beekeepers, especially those who want to use, your human health is more important than bee health, I would say first. So be careful when you use a vaporizer. You have to use a proper gear, like Carolyn is wearing a respirator here. And this is the device that we used. And we were struggling to use what kind of device is more popular. So we did some digging and thought this might be the best one that probably can relate to most of the beekeepers that use this kind of technique. So here is how it is put in. So we use a spacer, and I know many beekeepers probably don't use a spacer. I would recommend use a spacer, because when you're putting this in, I have seen some people burn their bottom bars of their frames because it gets hot. So if you're sticking this thing, this wand in without a spacer, then yeah. And again, if you want some sort of a uniform spread of these crystals when they vaporize, you need to have some space. So please try to use spacer if possible. So for this study, we used a spacer like this. So we have a master beekeeper program as well, which we started in 2011. So some of those master beekeeper students wanted to be a part of this science. So we involved them to get some research experience. So you can see some of those master beekeeper students also assisting as they were well trained before they started this task, so that our data is not compromised. So here you can see we put these acetate sheets. Probably some of you are aware of those. These are transparent sheets where you can see through. And then we marked all these larvae of different ages. So we were looking at 2-day and 4-day old. So that's what we wanted to look, at least. Maybe we could have done all ages, but that becomes a more laborious task. So we looked at age groups. Young would be 2-day old, and older would be more towards 4-day old. So we gave them different marks here. You can see in the next one here, I don't know if you can see, there are some dots here. So we picked 50 larvae of each of those age groups from each of those hives. And then we continuously monitored their development, right? So that's how you look for, if there was an egg or a larva here, I go on day 2, and if it's missing, then we mark it as it's gone. So that's how we monitored the different developmental phases of these young larvae, old larvae, as well as eggs. So here I have quick results here. Again, I have to analyze in different ways, but I just put it just for some simplicity here. So percentage of eggs lost here on the y-axis, and you can see after 24 hours. So your formic is the yellow one, and your dark, whatever you see, the black bar here is your oxalic, and this is your control. So there was no significant difference between control and oxalic, at least after 24 hours. But there is a significant mortality you can see for eggs, about 32-33%. This is after 48 hours. And then after a week, if you look at those same frames, you're losing about 90% of the eggs in formic. And again, you may ask what, so this was 2 pads for that 14-day period that is recommended. It's not the single pad dose. So here you see, again, no significant difference between formic and the controls. So here I'm showing you the fate of young larvae here. So you can see again, on the y-axis is the mortality or the larva lost. You can see after 24 hours here, I've changed the colors. So here you see the beaver color orange, here Formic Pro, about 20% loss. Here, don't see a significant difference between oxalic and controls. Same similar trend after 48 hours. After a week, you're losing about 70% plus for Formic, and about So you may be surprised, why is there loss in control? So I will try to discuss at the end if you have time, but probably it's not fair that you assume that all the larva that is there in a hive will survive to their maturity, right? So there is natural mortality for some other reasons as well. So that's probably an explanation why you see mortality in some control as well. You should not expect all 100 eggs laid in the next couple of hours will survive and become adults. So here is the fate of old larva. So old larva looks like, you see the scale is different here. So only it's a lower, so about 5% damage for the 24-hour period for the older larva. So it looks like older larva are a little more resilient when compared to the younger larva or the eggs. So same you see after a week, you have this Formic Pro about 25% loss. But there is, again, look at all these graphs, there is no significant difference between the controls and oxalic, at least for that one dose that we applied. So here is the efficacy one that I warned you not to glean too much from this, because it's really not an efficacy study when compared to oxalic, because oxalic one treatment doesn't do anything. You can see here, actually it's worse than your control after 3 weeks period, what you look for here. So here is the one when we started looking at this, and then here is the dotted line is where the treatment went in, either oxalic or formic. And then you can see the formic really, again, I'm preaching to the choir. You all know formic really works and it's a very effective product. So there is no question about the efficacy of formic. You do get a very good control, but the only thing is you have to be careful how much brood you want to sacrifice for. And so it's about the right time. If you're using at the right time of the year and when you still have a scope of increasing your population. So not try to use late like August or September, especially here in Oregon or parts of Washington, because then you don't have much opportunity of your colonies to grow big, because you need those really good winter bees going into winter. So maybe sometime you have to find that or the sweet spot where you have to use formic so that you can get rid of mites as best as you can and still not deplete your population of bees. So take home message, oxalic looks like, at least with our study, doesn't appear to do much damage. So it appears to be safe. Again, it's only the 1 gram per brood chamber dose. That's what the recommended is. But I heard from, at least from University of Florida, they're thinking that 1 gram per brood chamber is not the right dose. Maybe it should be higher than that for getting a control. But again, I have no idea. We tried 2 grams. So when I speak to you group next time, I will have data on that. So we use 2 grams this year as well, and repeated another study, not looking at larval toxicity this time, but looking at the efficacy of the product, both formic and oxalic, with a 2 gram dose as well. And we used actually even 4 grams. So we tried 1, 2, and 4, 3 doses. So yeah, I haven't analyzed any of that data yet. So hopefully next year, if you're interested, you can email me in December. I may have some data on that. So formic acid, as I showed you, has negative impact on the brood, but it is very effective in Varroa control. And I think some of you asked, probably Randy was answering some of those questions on if formic is and killing. So we have used, again, that's out of context I'm talking here, but the single dose, like the single pad doses, we haven't seen any impacts on queens either. So the queens, I think there was a discussion on how I think it was Jennifer Short and Randy probably discussing on what's the mode of action here, or how does the queen gets damaged. Yeah, so Randy is right. It's not the oxalic or the formic that directly kills the queen. It's probably other indirect effects. So what I can think of one more is the feeding, right? The queen needs a lot of protein when she is laying eggs, and so when there is a lot of disruption, we think, in some of the cases we have seen queens are hiding somewhere in the corner, or they go onto the bottom board as well if the fumes are too high. So maybe all those things, maybe the feeding of the queen, so the queen is not being attended properly, probably. So again, these are speculations. I don't think anyone has done really a concerted study to find if what happens, what's the mechanism, why does it impact queens. But I do hear from commercial beekeepers as well as hobby beekeepers often that sometimes they lose 10-20% of their queens when they use formic. So yeah, we really don't know the mechanism, but again, a speculation to add to the points that Randy and Jennifer made this morning. Maybe there is something about the queen not being attended because the bees are in a disarray, so they're not attending the queen. So the queen is on her own for a significant amount of time, maybe a day or 2 or 3, which could impact queen health. Okay, the last one that I promised to discuss is this Varroa treatment option, which probably most of you use, APVAR or Amitraz. So Amitraz efficacy, or I know I hate to use resistance, but again, just to give you some sort of an idea what we are looking for, I have used the term resistance as well. So we have all these options available for beekeepers, but this is the most popular one, at least for the last 4 or 5 years since the registration was obtained for Amitraz. And before that, I know people have seen this product, I guess. Uh, uh, this was used by commercial beekeepers and many others in the past. But again, I don't have any concerns about beekeepers using it because I think it has saved the industry. At least the commercial beekeepers have relied on this product for a long time because there was no registered product available until last 3 or 4 years before, right? So what's the reason why we wanted to look at this problem? Like, why would you want to look at Amitraz resistance? Is there a resistance at this time, or is it not working? So we thought the off-label use of Amitraz, because beekeepers have been asking this question for a long time, right? At least for the last few years, that is Amitraz working? So are mites, are we getting good control using this Amitraz? That's a basic question. So we wanted to see how this can be addressed by sampling a large number of hives and using a PETIS test. So these are the reasons: off-label use of Amitraz, use of different dosages. You talk to commercial beekeepers and they don't have one protocol, right? They are using in different ways, different doses, different frequencies, different formulations probably. So this is all, again, in last few years, they were not even able to obtain something, a legal product, legal in the sense that is sold in the US. So we don't know what's going on with that problem. But again, I have to point out that we have been very lucky at this point that we are not seeing any Amitraz resistance problems as far as my knowledge goes. So few studies have alluded to this, but again, not conclusively documented the Amitraz resistance. There was one recent study that I saw from USDA lab, Frank Rinkovic. So he did some studies both in the lab and the field. And again, it's still very an open question. We don't really understand what's going on in terms of resistance. So the whole idea for this study was that the beekeepers were very worried, at least about the efficacy of the registered product that they are using at this point of time, which is your APVAR. So we wanted to compare whatever I said, again, resistance or efficacy, the term, I'll leave that up to you to imagine. So we wanted to look in both the Canadian population and the US. The reason why Canada is, again, one of my colleagues here at OSU, Anthony Melitopoulos, he suggested Dr. Shelley Hoover in Alberta. So we wanted to collaborate with them to see, because Canada has been using the registered product for a long time. So we have been using only for the last 4 or 5 years, but Canada has been using longer. And they, as far as I understand, those beekeepers don't use the off-label product. So we wanted to see, maybe there might be some insights we can glean from their mite populations to see if they are getting the same control as we, or maybe we are not getting good control when compared to Canada. So we collaborated with them. And so this is a very brief description of the methods here. So PETIS test, probably most of you heard. So Jeff Pettis used this protocol since that Apistan, or even for Coumaphos, I guess. So we, again, because of lack of information and any other published studies, we had to rely on something that we knew was done before. So again, I'm not saying this is one of the best methods to test for this, but as I said, given the lack of information from past literature, so we went with modifying the PETIS test. And so it's about 1.5 inch APVAR strip. It gets stapled on an index card, and the index card goes and sits in that 500 ml glass jar. And then we release 300 bees from those hives for looking for mite control or mite mortality. So we used a 24 incubation incubation period. It may be too long, but we wanted to see what happens. That's a maybe that's a worst case scenario to look for. Maybe you'll get probably 90% mites off within 4, 5, 6 hours as well. So the dead mites were shaken, counted after 24 hours, and any remaining mites, we extracted them by putting alcohol and then counting how many were remaining. So that's how you count for the live mites. So here are wonderful people that helped with this study. So we went in 4 different trucks to this area called Madras in Central Oregon, which is a carrot seed pollination. We get about probably 15,000 hives for pollination there. So this is a place where you can get too many hives that you want to do this kind of study. So we sampled over 270 hives, and sometimes the time it's 24 hours, right? So sometimes the students had fun doing in a park as well, sitting and doing, because you have to do those counts exactly at 24 hours. So here is a very basic information that I wanted to share. So here you can see, this is the pink one is your United States. Here is the Canadian data that we have. So we didn't see a significant difference in the Varroa mite mortality. So it doesn't appear that we have a mite population that is really resistant or is not being controlled at this time. So that's what we found. Again, we are still analyzing this data for different interpretations, but this is something basic that I wanted you to learn about this. So this is the interesting one. I want you to ponder on this one. So for example, we suppose these are 6 or whatever, 7 operations that I have listed here. This is beekeeper A, beekeeper B, C, D. So if you look at those, the ones I talked about, 270 different hives. So if suppose beekeeper A, we sample whatever 40 hives, then if you do their average mortality for those, you get about 91% control, which is pretty good. I mean, the mites are still being impacted by APR or the emitraz, but then there will be some outliers, maybe 1 or 2 hives that get only 50% control. So the mites are still alive after 24 hours of emitraz exposure. So that's what I don't understand at this time, and this is still a puzzle. And even that paper that I referred, Frank Rinkovic from USDA, I think he had similar observations as well. And so here we don't know, maybe there is more work to be done actually with those vial assays in the lab, and maybe some more robust studies need to be done to really understand what's going on. But just with this study, I can't really say if it's a resistance problem or is it just one-off hive where we are seeing these mite populations that are not being able to be controlled with APR. So same thing you can see again, I'll give some examples here with 87%. If you look at those all, suppose beekeeper C, we sample 50 hives. And if you do an average for all those 50, you get a 87% control. But then this has come down to 87 from whatever 90s is because of 1 or 2 outliers like this, where you're getting only about 56% control. So that's still a puzzle which needs to be solved. So I'll just go over some points I want to make, especially again for the new beekeepers that really in Varroa. Again, I'm glad Randy, Jennifer, everyone has at least highlighted some aspects of Varroa. And I don't think you should get tired hearing about Varroa, because this is the major one that you have to deal with in your operations. And this will make or break whether you're a beekeeper or not down the line. So be very careful about Varroa, and yeah, do as best as you can to control. And I know you're in good hands with your beekeeping groups in Washington, and please be mindful, keep monitoring. So that's what I say here, continuously monitor mite levels in your hives. Monitor mite levels before and after treatment. This is something I know it's very known fact, but sometimes we ignore this. So if you are using a formic or oxalic or APR or whatever your product is, I would always ask you to go back after 3 or 4 weeks and do another mite sample. Uh, as Randy was saying, alcohol wash is the best method still. And again, with shaking and all, there might be some variability, but please use alcohol wash and look before and after levels of your treatment. Because if you go after 2 months and look for your treatment, if it has worked or not, you have wasted your time, right? So there is no more going back. So be careful of that, be mindful, and try to do mite monitorings before your treatment and after, so that you know for sure your treatment is working. And even a 2-week delay, don't take them easily. I mean, I have seen some commercial operations where they have 10,000 hives, so they can't get to them all in time. So I have seen even yards that are 2 weeks late in my treatment, they do suffer. And because these mites, again, viruses are the major problem, right? So be mindful about that and try to get the treatments as soon as possible before the damage is done. Because those winter bees that you are planning on having in your hives that will come back and in spring and forage, those need to be as best in a condition as possible in terms of both Varroa mite infestation levels and nutrition. Those are the two critical ones. You can't ignore those. If you have good control on both Varroa levels as well as good control on nutrition, I think probably your success rate will be high. So colonies, I get this question from hobby beekeepers a lot in Oregon. My colonies look great. I looked in October, they were booming with bees. I had probably 16, 17 frames of bees full, and they were just overpowering. And then I went in December and most of the bees are dead, or I don't see any bees in there. So this again is like just looking at the bees is not a good way, right? Especially if you're a new beekeeper. Bees may look good. I may look good today. If I had any virus like COVID or something, I may still look good to you. But if my immune system is compromised, it's the same way. Your bees have a compromised immune system because of high mite loads, and they have transmitted those lethal viruses already. So then they may look okay to you in terms of their physical fitness. But again, when the time comes, their immune system is so compromised, they will all die by December. So that's what happened. So again, I can't emphasize more on Varroa control. So please be mindful of this and try your best to control them in time. So this is again a Honeybee Health Coalition website. I just wanted, if you want to get more information, you can go there and look for some of those videos on how to apply these treatments. There is a lot of good information in there. Okay, I think I have left enough time for questions, and at least 10 minutes. So I'll be glad to take those questions.
Thank you, Ramesh. Yes. There's one question that I see from Julia Common. Have you considered doing a trial using formic acid on meat pads? Less intense for bees, resulting in not so much disruption and hence good queen survival, and less interruption of laying, although more work for beekeepers.
Yeah, great suggestion, but unfortunately, no, I haven't. I am just struggling to do only the available products that are right now for beekeepers. But yeah, I mean, maybe someone can do this in the future. But no, I haven't used meat pads, and maybe someone else has some insights on this. But yeah, it's all about, again, to address the basic point that you're trying to make. Yeah, it's a release, and those things are critical. How much vapors you have at a point of time, and you shouldn't have that. You should have the balance where you should be able to kill mites and not have too much of a damage on. I mean, you will have some damage, but I'm looking for something that is more optimal. So yeah, short answer, we haven't done, but I'll think about it if I have time in the future.
There's some good research out of Canada on that.
Oh, who did that? Do you know who is that one, Randy?
I was just reading the paper just last week. Shoot me an email and I'll send you a citation.
Okay, cool.
Okay, so Ramesh, first of all, yeah, you do look marvelous, whether you have viruses or not. So yeah, but hey, here's some more questions. Jaylene Naylor writes, my knowledge is likely not up to date, but I thought that serial treatments using OAV, which I now know what it is, treating multiple times every few weeks was not approved by the government agency that approves that thing, whether it's EPA or whatever you have in Canada. Do you know anything about that?
Yeah, good question. I have to, maybe Randy, you can correct me. I can't recall the label exactly now. I know you can't
I'll
use
tell you right now. It says spring, early spring and late fall treatment only.
Yeah.
And it also says you cannot do it while this honey
supers are on. Yeah.
Okay.
Yeah, so
can you do it more than once? Can you do multiple treatments every few weeks?
Yeah, that's what we don't know. That's what I said, right? Because we wanted to see what beekeepers are doing right now. But yeah, the one treatment, as I showed you, you will not get your mite control that you want. So that's what beekeepers I have seen use 4 or 5 times as well. So we wanted to see. So that's a great question. So the next time we want to look at 3 different applications. So this year actually we did that. So I'm showing the data from last year, but to 2020 we did 3 treatments. So I'll have that data available soon. So we
Okay.
wanted to see if it's more toxic. I know people are really worried if we hit 3 times a week apart. And I know there are beekeepers who are doing that already. Even some commercials I've seen hitting every week for at least 3 weeks straight. So we don't know what kind of damage. I'm just showing you the label dose, 1 gram per brood chamber and only 1 time. But again, yeah, the jury is out still on the other parts of the study.
In my experience, I don't notice adverse effects with even with 10 treatments, 10
Okay.
days apart. But it takes a lot of treatments to get the mites down during the summer.
But Randy, do you use a 1 gram dose or
The 1 gram per brood chamber, but 2 grams per double
per hive? Yeah.
brood chamber.
Yeah.
I am not a big fan of oxalic acid vaporization. Boy, I'd like to show you if you're, I checked when you came up with those figures, Ramesh, of the percentage of mites in the brood in September.
Yeah.
I go, wow, some great data. Let me check it on my mite model. It came up exactly the same number, my mite model.
Cool. Okay. Yeah, I wanted to share this with you at some point because we just started analyzing this. So, but I'll send you some raw data if you're interested in your model to see.
Oh, of course,
Because
yes.
we have all these 6 hives. I know the exact number of mites we found in each of those hives as well. Yeah.
Beautiful.
Nice. Yeah, so the other 2 questions actually that are on the Q&A are related to this. One of them says, Sagili and Randy, I guess formal and informal descriptions. Your opinion, please, of fogging with oxalic acid once a month, then in the fall, once a week for 4 weeks in a row. So I think you've answered that, but that's the question.
Yeah, maybe I'll allow Randy. I think he has used it personally, but I only tested once. So you understand, Randy, with that?
Yeah, we use a lot of oxalic acid in our operation, have for years with the dribble. And now we've done several years worth of the extended release oxalic acid. And then last year, I wanted to test the vaporization and got the crew involved, and we played with it for a while. And everyone on the crew said, why in the world would anybody use oxalic acid vaporization? It takes more time, it's more expensive, it's dangerous to the applicator. And we all decided, oh, it must be because those beekeepers don't want to ever crack open the colony. Other than that, we could not see any advantage to using it. Now during the winter in Canada, where the fall comes on very suddenly, and they don't want to crack their hives open and break the propolis seal, well then, and they're broodless, it makes complete sense to use oxalic acid vaporization there. But during the warm weather, and Ramesh, I'm glad you showed Carolyn with the protective gear on, because it is really easy to inhale oxalic acid. It's not actually a vapor at that point. It's called a vapor, but that's not a vapor. You can't see oxalic vapor. Oxalic acid vapor is invisible. The fog of oxalic acid crystals that you see, and you've inhaled that. The problem is unlike formic acid, if you inhale formic acid, your breath will go in and go and stop. Your body won't let you inhale formic acid because it's irritating immediately to the membranes in your throat. Those crystals of oxalic acid, you can take a full lungful before you feel anything, and then it's way too late to do anything about it.
Yeah. The only thing I will add for that is like the 2 gram dose. I don't know. The University of Florida folks, they think 1 gram is not. I mean, you're right, Randy. There is. People are not happy with the current dose that is on the label, and I don't think people are getting, like, even our study, like you saw, in 1 week there is nothing. Actually, even the controls look better than the oxalic. So we don't know. Maybe the dose is an issue. I don't know. So maybe we tried both 2 grams and 4 grams, which are off label for now. But I think I don't know if EPA is interested in that, or sorry, USDA, whoever wants the data. But it looks like maybe the dose is an issue as well.
My feeling so far is not the dose, but it's the duration of the exposure. And this is what I'm working with the oxalic and glycerin. And I'll be talking about it more tomorrow, but when I put in the oxalic acid and glycerin and then remove the pads at the end of the season, the total amount of dose that actually goes onto the bees is tiny. It takes very, very little oxalic acid on the bees to get high efficacy if it's in there continuously. So as the mites are coming out of, like, if you vaporize it, if after 3 days there's no more effect. So that's you've got 12 days worth of mites in the brood. So you're getting 3 days worth emerging that might possibly be affected, probably about 2 days worth. And then the other 10 days worth have no effect at all. So it's not so, and